The mouth will first break down the food such as meat, and it is coated with saliva, which is made up of water, mucus, and hydrolytic enzyme called salivary. Our teeth and jaw muscles help crush and crumble the food. Then it will travel from the mouth through the esophagus. The food bolus reaches the end of the esophagus and arrives at the cardiac sphincter into the stomach.
Chemical digestion begins in the stomach because the stomach will secrete pepsinogen, which will then convert to pepsin after secreted. Pepsin is the one that hydrolyzes the peptide bonds. This means it breaks down the amino acid bonds. To explain a little further, stomach cells only begin secreting pepsinogen in the existence of food, and at the same time, hydrochloric acid will be secreted in order to assist in breaking down the food. So it explains that the hydrochloric acid is the one that starts the conversion of pepsinogen to pepsin, which then is able to hydrolyze the peptide bonds, breaking down the protein to polypeptides.
Then it will move on to the small intestine, which completes the chemical digestion of proteins. The duodenum is major chemical digestion and absorption of proteins. The pancreas begins to secrete some enzymes that help digest proteins. From the pancreatic juice, an enzyme called trypsinogen is released. Trypsinogen converts into trypsin. Trypsin is basically the active form and will hydrolyze the peptide bonds, meaning it will break down the amino acids that are linked together. After this process, polypeptides will be digested into smaller
Polypeptides further, so our body is able to absorb them. The very last part of digesting the protein is by the brush border enzymes. They also hydrolyze peptide bonds, so these polypeptides will be digested into tri- and dipeptides and amino acids. Then our body is able to absorb these. They are absorbed through the co-transporter with hydrogen, and the proteins, the smaller form polypeptides, can be hydrolyzed by cells, which will then break it down into amino acids. Now, the amino acids can spread into our bloodstream where it will be carried to the liver by the hepatic portal vein. The amino acids that don't stay in the liver are transported to the rest of our body to be taken up and utilized by other cells. The waste then goes through the large intestine where the reabsorption of water from the indigestible matter occurred. Then it is stored in the rectum and released through the anus. The mouth will first break down the food, for example, bread, and the food is coated with saliva, which is made up of water, mucus, and hydrolytic enzyme called salivary, which functions to break down starch into smaller chains of glucose called maltose. Our teeth and jaw muscles help crush and crumble the food. Only about 5% of starch is broken down in our mouth. Then it'll travel from the mouth through the esophagus. The food bolus reaches the end of the esophagus and arrives at the cardiac sphincter into the stomach.
When carbohydrates go down the esophagus into the stomach, no chemical breakdown occurs because the amylase enzyme doesn't function in the conditions of the stomach. The stomach will only mix the content around and allow the starch, the carbohydrate, to reach the small intestine. But the peristaltic contrast of the stomach mix the carbohydrates into a more uniform mixture of chyme, which is a mechanical breakdown.
The chyme is expelled into the upper part of the small intestine. Before entry of the chyme into the small intestine, the pancreas releases pancreatic juice through the pancreatic duct. The pancreatic juice contains the enzyme called pancreatic amylase, which break down the starch into maltose. Maltose is essentially two glucose molecules linked together. Enzymes are also secreted by the intestinal cells that line in the villi. Maltose helps break the bond between two glucose units of maltose, and another enzyme called lactase breaks the bond between galactose and glucose. So through starch digestion, we will end up with many glucose molecules. There are also many sodium ions that play a role in the absorption of glucose into the body. Once all the carbohydrates are chemically broken down into single sugar units, they are then transported into intestinal cells.
The cells in the small intestines have membranes that contain many transport proteins to get the monosaccharides and other important nutrients into our blood, where they're able to be spread out to the rest of the body. The first organ to receive glucose and galactose is the liver. The liver will take them up, and galactose converts to glucose and stores glucose as glycogen or exports it back into the bloodstream. The waste goes through the large intestine, where reabsorption of water from the indigestible matter occurs. Then it is stored into the rectum and released through the anus. The mouth will first break down the food, and the food is coated with saliva, which is made up of water, mucus, and hydrolytic enzymes called salivary. Our teeth and jaw muscles have crushed and crumbled the food. Through this process, fat is able to become tiny droplets and separated from watery components, then will travel from the mouth through the esophagus. The food bolus reaches the end of the esophagus and arrives at the cardiac sphincter into the stomach.
Inside the stomach, gastric lipase begins to break down triacylglycerols, which are the simplest lipids formed by fatty acids. When the stomach contracts, it helps disperse the fat molecules, but during this process, very little fat digestion occurs because fat tends to clump together and form large droplets as it moves through our digestive system. Also, the pancreatic juice, entering the duodenum through the pancreatic duct, releases lipase, which makes fat break down into 3 fatty acids and glycerol.
When it reaches the small intestine, fat isn't digested at all. The digestive system manages to combine the separated fats with the watery fluids created, which is bile. Bile contains bile salts and substances derived from the cholesterol, so it stabilizes processed foods. Bile salts break down the fat into smaller fat droplets. Bile attracts and holds onto fat while it is held onto by water. This process of emulsification increases the layer of lipids over 1,000-fold, making them more accessible to the digestive enzymes. The finer droplets now have more surface area, and this aids digestion because the pancreatic lipids can only act on the surface of the fat droplet. The enzymes in the small intestine are now responsible for the most of the fat digestion. When the pancreatic lipids act breaks down the lipids, it creates free fatty acids and monoglycerides. The fatty acids and monoglycerides give little trouble being absorbed out of the digestive tract.
The absorption is in the mucosal lining along the small intestine. Once they are in the cells, the free fatty acids and glycerides enter the endoplasmic reticulum, which is able to transport the lipids. In here, they are coated by protein, which protects them from water. Adding on, the lacteals is another way fats are absorbed. Now they move into the hepatic portal vein, into the liver. Now fats are able to move around the body. They can build cell membranes and make myelin sheaths, which coat our nerves and insulate the body with the fatty cushions. They are also used as a fuel source if we don't have enough carbohydrates in our body. However, if fats aren't absorbed properly, a person's stool will contain high amounts of fat. The wastes go through the large intestine where the reabsorption of water from the indigestible matter occurred. Then, it is stored in the rectum and released through the anus.